Okay, I'm back. So, I wasn't exactly able to complete this in one video. That's fine. This is the second video. And I left off with the G stimulatory pathway that activates cyclic AMP, protein kinase A, and activates CREB basically and causes transcription of genes, or that activates other downstream effectors, say in the liver or any other kind of cell type that are more immediate, that can contribute to, say, release of glucose from the liver and production of more glucose, or storage of glucose in your muscles, or even relaxation of smooth muscle in your muscles to give them more blood in the fight or flight situation if your sympathetic nervous system is being activated. I'll talk about a little bit about with respect to dopamine, for example. Dopamine in your brain, for example, you have this would be a D1 receptor, one of the versions of the D1 receptor, and this would be like one of the versions of your D2 receptor. In schizophrenia, D2 is being inhibited, so you're inhibiting the GI pathway, the inhibitory pathway that antagonizes the stimulatory pathway and stops cyclic AMP and protein kinase A by trying to inhibit adenylate cyclase. You're stopping this pathway, so it's actually activating towards the D1 pathway, at least if it's just binding to D2. So in your basal ganglia, this would be activating, and I didn't talk about this, but maybe I will in a different video. If you're using first generation antipsychotics, you're basically activating your indirect pathway, but inhibiting your direct pathway by not allowing dopamine to do what it wants to do by binding to the receptors, which isn't that great of a thing. It kind of causes Parkinson's-like symptoms. I think causes too much GI function in the basal ganglia, or not enough of it, I guess. So your cholinergic nervous system, I think, ends up more activated. Drugs like benzotropine and procyclidine, those kinds of drugs, I feel like I'm forgetting one, bipyridine, those drugs help to modulate this through muscarinic receptors, because you also have the muscarinic receptors. So basically what I'm trying to say is that dopamine is actually another catecholamine, and it also affects these receptors, and even antipsychotics operate through this pathway. Your locus coeruleus, for example, mostly releases norepinephrine into your brain, and your hippocampus, your forebrain, and different places in your body, even down into your spine. And when it binds to the alpha-2 receptor, which is through the GI pathway, it can help inhibit some of your pain sensation. But in your hippocampus, for example, sometimes that norepinephrine is not actually converted. It's not actually converted to norepinephrine, because in your pathway to norepinephrine, you end up with dopamine, epinephrine, or dopamine, norepinephrine, and epinephrine. Well, sometimes before it even gets converted to norepinephrine, that dopamine can also be put into those vesicles too, at the terminals from the locus coeruleus, and it can secrete dopamine instead, and that actually helps aid in memory. So dopaminergic signaling is important and rather interesting in the brain. Antipsychotics inhibit this and kind of aren't exactly the best thing to take if you want to be intelligent or think, So just a thought. And they used to do lobotomies and cut parts of people's orbital frontal cortex out, but I'm not going to talk about that in this video, but it's kind of interesting. So I talked a little bit about GS signaling, but I did not talk about the negative feedback loop on this. So let's talk about that. So if you have a bunch of cyclic AMP, it's activating protein kinase A, it's activating CREB, and CREB has longer term effects, like in the hippocampus when I was talking about, say, dopamine, for example. If it's activating the GS pathway, then it's activating CREB, and CREB is important in memory. It can help with memory as well, along with CAMK2, which I'll talk about later. So anyway, there's a negative feedback loop. This is what's called phosphodiesterase, this pink here. Phosphodiesterase, when it's activated, which it can be activated by protein kinase A itself to negatively feed back, destroyed that, to negatively feed back on protein kinase A, and that tends to happen in cells, which is kind of a good thing, right? When you regulate and don't have too much of something. Cancer is not a good thing, right? Anyway, so in this case, though, it's activating phosphodiesterase. Phosphodiesterase actually converts cyclic AMP back to adenine 5 monophosphate instead of the adenine 3,5 monophosphate. So it cuts the cyclic bond, so it's just connected to the 5 position. I'm curious what happens if you just cut the position of the 5 and just end up with the 3. And I actually looked it up, and it does different things in the cell. So you can have different isomers even of the cyclic or even of the monophosphate version, which would be interesting. And you could actually theoretically have different isomers of ATP, like these 3. What if you connected them to this 3 position here or the 2 position, right? Like biotech companies should really think about that and try to figure out if there's a way to make a energy substrate, like some kind of energy storage, and just use different enzymes to use it to actually activate the specific pathways you want to activate in the cell, which would, well, you'd possibly figure out how to make mitochondria make these molecules too. But if you can figure that out, later in human evolution, when we bioengineer ourselves, maybe that'll be a way to change things. Anyway, going back here, this is phosphodiesterase. I think there's a version of it that's connected to protein kinase A at the beginning, and I think it might be active even at the beginning, helping this to be an all or none response. So you have to have enough cyclic AMP to actually activate protein kinase A, otherwise this thing is gobbling them up, even though kinetically it's kind of weird, kind of hard to understand, but remember that these A caps, they can bind to certain places in the cell, and so that would actually spatially confine where the cyclic AMP molecules can come from to activate protein kinase A and bind like at least two for each substrate. So like in the heart, I think they're, I don't know, I can't remember, I think they're by the ER if I remember right, but I'm not 100% sure. But it depends on what cell type. This can be confined to different places in the cell. Even the position of phosphodiesterase can determine how well protein kinase A gets activated. Protein kinase A activates phosphodiesterase. Phosphodiesterase inhibits the signaling that's going on and helps to bring us back to AMP so that we can stop the response. Also, protein kinase A can go and phosphorylate the receptor, the stimulatory receptor, like the beta-1 receptor, or let's just talk about the beta-2 receptor. The beta-2 receptor, protein kinase C, which I haven't talked about here, which I will later. Protein kinase C can also feed back from the GQ pathway. Oops, yeah, my bad. Protein kinase C can phosphorylate this. There's also other proteins called G protein coupled receptor kinases, which I guess would include these. I think there might be other ones that phosphorylate the beta-1 receptor, and in doing so, prepare it for other proteins like beta-arrestins to bind to the beta-2 receptor, prevent signaling, but it has to be bound to ligand. So if there's enough ligand out there, it can definitely help this process occur. It can stop the G protein coupled receptor signaling, and then clathrin and I think AP2, which is another another complex that can bind. Basically, the result is clathrin-mediated endocytosis into endosomes and then to lysosomes, and you can chop up this receptor so that you don't have a lot of receptors anymore, and it's a way of it's basically a way of desensitizing the beta-2 receptor. I think it also occurs possibly in alpha-1 receptors. When I read a review, I think they suggested the possibility of that with alpha-1 as well, which is actually GQ, but that might also happen there. So I've just talked about the G stimulatory pathway. We'll move over to the GI pathway here. Oh, by the way, GS helps increase chronotropy and ionotropy in the heart. So, and I didn't talk about that. I guess I'll talk about that real quick. Protein kinase A can also phosphorylate some of these proteins in the endoplasmic reticulum, including phospholamban, which I don't have drawn here, and L-type calcium channels, and also the sodium calcium 2 plus exchange proteins in the membrane, which allow you to pump out calcium even faster, and in doing so increase lusotropy, which allows the heart to relax faster. So during diastole, it can actually take more blood, but then it also enhances the function of the calcium channels, so calcium pumps in real quick, causes contraction, and it's even contracts even more. And in your sinoatrial node, this may lead to, like, you have these receptors called hyperpolarization activated cyclic nucleotide channels, cation channels that can bring in mostly sodium, not as much potassium. I'm not sure about calcium, but it brings that into the cell and helps reduce the amount of time for phase 4 depolarization, which can increase your basically increase your heart rate, chronotropy. There might be other effects, though. I'm not sure how much of it, though, is actually from the cyclic AMP, because I saw a paper that suggested it only just affects how high chronotropy can go or how low chronotropy can go. So you have to look that up yourself. So I just talked about G stimulatory pathway. GI, this brown here, starts out as a trimeric protein. The beta and gamma subunits here, those dissociate, go over, open up your potassium channel, which caused hyperpolarization. Hyperpolarization, since you have potassium leaving, I guess if you just want to think about it in terms of charges, even though how many potassium molecules actually leave, I don't know. It only has to be enough for it to actually reach equilibrium even, but even that makes a huge difference. It doesn't even take that many to leave to make a difference, but when it does, that hyperpolarizes the cell, so that makes it less likely for any kind of voltage-gated sodium channels, for example, to open up if it's in any other part of your brain. Like in this case, this would be like muscarinic 2 or 4 receptors, or any other G protein coupled receptors that are inhibitory, and there's a lot of them. You know, like there's 350 at least that aren't olfactory. And then, so the other effect is your alpha subunit, the GI subunit, goes over to adenylate cyclase. It inhibits it, so you stop making cyclic AMP from ATP. This will probably be good in situations where you need to conserve your energy. You don't want to use too much ATP. And in situations where you want to halt any kinds of effects of the stimulatory pathway as a negative, as an opposing effect to the stimulatory pathway, okay? Those are two effects of that. Let's see, GI, I mentioned muscarinic 2 and 4, and also there's the alpha 2 receptor. And in doing so, for example, you have the alpha 2 receptor in your brain, like in your medulla oblongata. It can, when activated, agonized, if we're activating the GI pathway here, in your medulla oblongata in certain nuclei, which I think are downstream of the hypothalamus somewhere in your medulla oblongata. In doing so, that actually can reduce the amount of the effect of, say, this pathway in your vascular smooth muscle all throughout your body, which is activated by norepinephrine. So it's kind of a negative feedback loop on your sympathetic nervous system when you activate this pathway in the medulla oblongata in certain cells using your alpha 2 receptor. Also, another example, the alpha 2 receptor drugs like, guanfacine. Guanfacine is also used for ADHD, and I can't figure that one out, but I guess it works. And it also decreases blood pressure. I'm not sure how that would work with, like, say, D-amphetamine with salt, but I would think it'd probably be pretty good because it lowers your blood pressure and also helps with ADHD. That's just like a rabbit trail. But in inhibiting the alpha 2 or in activating the alpha 2 receptor, say like in your pancreas, for example, like, I didn't say all the drugs yet. There's dexmedetomidine and Guanadril, methyl dopa, which is mostly just used in pregnant women for high blood pressure, for hypertension, and clonidine, which is also called catapress. Those are on the alpha 2 receptor, which actually I shouldn't draw blue. Let's see. Alpha 2. Like in those cases, you're activating this pathway, causing hyperpolarization of those neurons. I take it somehow they're either activating or they are neurons that cause vascular contraction by secreting norepinephrine to these smooth muscle on the alpha 2 receptor. And how that's laid out in space, I don't know, but I just know that this is inhibitory towards high blood pressure and vascular tone, and can help with ADHD if it's at least guanfacine, but who knows where that is in your body. In fact, drugs like Strattera, they increase norepinephrine signaling, but maybe there's other places where it doesn't, and somehow inhibiting norepinephrine helps, even though I can't figure that out because cocaine inhibits the norepinephrine transporter, and that works in like, how many minutes or whatever, whereas Strattera is supposed to take like weeks to work. So I don't know what to think about the monoamine hypothesis, but you can figure that out on your own time. Moving over to GQ signaling. This activates calcium release from the endoplasmic reticulum. Also activates your store-operated calcium channels, causes calcium to come into the cell, into the cytosol. So initially, in this, let's just say the alpha 1 receptor. So in the alpha 1 receptor's case, say you have shock, they give you methoxamine or phentermine to help offset the effects of bradykinin and histamine, which are causing, maybe activating this pathway or other pathways that cause lower blood pressure, trying to relax your smooth muscle. They give you this, which is an agonist of this. This is going to activate, or in the case of norepinephrine, going straight to your extremities and activating your alpha 2 receptors. This trimeric G protein, you end up with dissociation of the beta and the gamma subunits. Your alpha GQ subunit goes over, it activates this protein, which is called phospholipase C beta, abbreviated PLC beta. And in doing so, and activating it, it's going to want to cleave the bond between diacylglycerol and there's two like fatty acid groups that go into your membrane to keep it anchored here. In doing so, it's releasing this molecule here, which is has phosphate groups at the 4, 5, and 1 position. This is abbreviated PIP2, PIP2. And what that stands for is phosphoinositol 4,5 bisphosphate. So there's actually, even though it says bisphosphate, there's actually 3 phosphates, but since you already said phosphoinositol, we're talking about the other 2 phosphate groups, so that's why it's a little confusing. That becomes IP3, which is inositol 1,4,5 triphosphate. This one has no hydrophobic groups on it. In fact, each of these parts of the ring has either an OH or a phosphate group on it. And this would, I think, be negatively 3 charged. So it's just going to fly through your cytosol and go all over the place, interacting with any water molecule it can interact with, until it finds your endoplasmic reticulum or your sarcoplasmic reticulum. It binds to the IP3 receptor. And once it binds to that, it causes a conformational change that causes the release of calcium, which you have a higher concentration of calcium inside your ER and your sarcoplasmic reticulum than in your cytosol. Your cytosol only has like 10 to 100 nanomolar, which, so we're looking at a really small amount here. That's like 1/10,000 to 100,000th of a mole, of a molar of calcium inside your cell. Whereas in here, we're looking at values that are 10 up to like 80 times as much if you're looking at the lower value, or let's see, like up to 8 times as much, or even down to just only 8 times as much. You have more inside your ER and your SR that are being released by both the IP3 receptor and also, not just for your IP3 receptor, it's also being released through your ryanodine receptor, which is activated by calcium that initially comes out. It also activates that. in doing so, it's causing protein kinase C to be translocated up to the cell membrane, and it's activated by diacylglycerol, which is the product that's left over after phospholipase C cleaves this. In doing so, it's activating protein kinase C. Protein kinase C can go down. It can inhibit the myosin light chain phosphatase, which is removing phosphates, and I might talk about this in another video, removing phosphates from your myosin light chain and promoting smooth muscle contraction. This pathway, the calcium, activates myosin light chain kinase by activating comodulin, and then you have, well, myosin light chain kinase is in the calcium comodulin modulated calcium comodulin dependent kinase family, and in doing so, it's causing that to become a kinase and phosphorylate what needs to phosphorylate, like your myosin light chain. It also, I think, desensitizes it a little bit if you keep activating this pathway. Calcium release, so that causes smooth muscle contraction. So this is your vascular smooth muscle. You're contracting those more distal areas when you don't need blood going to those areas, or even like your digestive tract. You don't need to digest, you need to fight or flight, like in the sympathetic nervous system activation example. In your parasympathetic nervous system, though, you want to be able to move food through your digestive tract, so you need to contract that smooth muscle. So there's other different effects of smooth muscle, like in your bladder, you want to increase the tone in that to cause you to pee, other things like that. So once the calcium level rises, some calcium binds to different proteins. It acts as a buffer. There's even dynamics where the waves of calcium traveling through a cell also have effects, like the frequency of calcium waves. There's proteins like calcium comodulin protein kinase 2, CAMK2, that's important in long potentiation of like your memory and your hippocampus, long-term potentiation. And that's a whole nother probably video to talk about, but basically that protein has 12 different monomers. It retains its memory once one gets phosphorylated. It can phosphorylate others inside of that, I think it's a dodecamer or whatever, but 2 hexamers basically. And that's like a memory in your body and your brain to remember that it got activated. And almost like 1 to 2% of your proteins in like your hippocampus and some of those neurons are made of this protein. So it helps it remember, and that's your memory part of your brain. There's a lot of research about CAMK2. But then also when a calcium level rises, you start to inhibit the ryanodine receptor and some of these receptors, and that's a negative feedback loop. Protein kinase C also deactivates by phosphorylating the GS, the beta 1 receptor, for example, the stimulatory pathway. It can also be negative feedback loop by phosphorylating this if ligands bound to it. So you actually have these different checking and balancing mechanisms depending on what cell you're looking at that help keep some pathways from becoming overactive versus the others. And then your GRKs can bind your clathrin, your beta-arrestin, and then clathrin-mediated endocytosis, etc. for this. Let's see if I missed anything. So protein kinase C inhibits myosin light chain phosphatase. Also, protein kinase causes smooth muscle relaxation by binding to or by phosphorylating the myosin light chain kinase and inhibiting smooth muscle contraction. That's why the beta 2 receptor might have effects on smooth muscle relaxation the way it does. So I think that's pretty much all I wanted to talk about. Thanks for watching this. If you like this video. Thank you.